Hello everyone, welcome to Prominent Biology classes. In this video we will discuss photosynthesis. As we all have some idea about photosynthesis, but at class 11th level we will study it in detail. So let's start the topic. Under the heading of photosynthesis, we will cover following topics: the introduction, early experiments, site of photosynthesis, pigments involved, light reaction, the electron transport system, chemiosmotic synthesis, C3 cycle, C4 cycle, photorespiration and factors affecting photosynthesis. Let's start with the introduction. Photosynthesis is a physio-chemical process. Physio-chemical means the light energy is trapped by the green plants and they convert it into chemical energy with the help of chlorophyll pigment. This is physio-chemical process. This photosynthesis has two importance. It is a primary source of food and it releases oxygen to the atmosphere. Plants, animals, we all need glucose as a food. So by photosynthesis, the plants make the glucose. And secondly, during photosynthesis, oxygen is released as a byproduct that we will see later on. To understand photosynthesis, many experiments was conducted by various scientists. Let's see one by one. Joseph Priestley, he conducted a series of experiments and concluded that air is important for the growth of plant. And secondly, he also discovered oxygen. One of the famous experiment that is given in NCERT is a bell jar experiment on page number 207, figure number 13.1. In this experiment, he used a burning candle and a mouse which is enclosed in a bell jar. Why bell jar? Because this experiment was conducted in a closed space. So after some time, he observed that the burning candle is extinguished and the mouse also suffocated. So by seeing this, he concluded that this burning candle and the mouse, they were somehow degrade the quality of air inside the bell jar. In another setup, he placed the green plant with a burning candle and the mouse, and he observed that this time the mouse stay alive and the burning candle continued to burn. So this shows that the plant prevent the degradation of the quality of the air inside the bell jar. Means because of the presence of this green plant, the air quality is maintained inside the bell jar. Another scientist was Jan Ingenhousz. The same experiment was done by this scientist. He experimented with aquatic plant. He took this aquatic plants in sunlight and he observed that small bubbles were formed around the leaf when it is placed in light, and identified that gas bubble as oxygen. He gave 5 points. Only green plants can release oxygen when it is kept in sunlight. So he concluded that sunlight is essential for the photosynthesis or essential for the growth of the green plants. The next scientist is Julius von Sack. In his experiment, he proved that glucose is produced by the green part of the plants, and this glucose is stored as a starch, and only green part of the plant can make the glucose. In his another experiment, he also proved that the green substance that we call now chlorophyll is produced in the spherical body within the cell, and that spherical body now called as a chloroplast. Means the chlorophyll was discovered by Julius von Sack. Our next scientist is T.W. Engelmann. Engelmann illuminated the green alga called cladophora, and the illumination is done by the prism. Means all the 7 colors are used to illuminate this cladophora, and this cladophora is placed in with the aerobic bacteria. Means this is the petri dish, and in this petri dish the bacteria is there, and the plant cladophora is also placed here. So what he observed, that the bacteria accumulated in blue and green light region. Suppose this is the blue light region and this is the green light region. So the bacteria accumulated here and here. And this is the first action spectra. He observed that the rate of photosynthesis is more in blue and red region. That is why this is called as the first action spectra. Our next scientist is von Neil. By the time of von Neil, it was known that the carbon dioxide and water is used by green plants to make glucose. Von Neil used in his experiment purple and green sulfur bacteria to prove that hydrogen is coming to reduce the carbon dioxide. That hydrogen is coming from the oxidizable substance to reduce the carbon dioxide into glucose. And he also said that the oxygen is coming from water, not from CO2. This oxygen is released in the atmosphere as a byproduct of photosynthesis. This oxygen is coming from water, not from CO2. And he showed this by using this green and purple sulfur bacteria. Here CO2 is combined with H2S. So after making glucose, this sulfur is released. So here the oxygen is released from water. This proves that the oxygen is released in the atmosphere is coming from the water, not from CO2. He later used the radioisotopic technique also to show to prove these 2 points. After an early experiment, moving to the next topic, that is the site of photosynthesis. We all know photosynthesis takes place in the green part of the plants. Suppose this is the leaf, and if we cut the leaf anatomy of leaf is like that. This is the upper epidermis, and above that the cuticle is there. And below this upper epidermis, the longitudinal cells are arranged, which is compactly arranged, and below this the loosely arranged some spongy cells are there. And these 2 different type of cells are collectively known as mesophyll cells. So mesophyll cells are made up of 2 different type of cells. The longitudinal cells, which is compactly arranged, that is called as palisade cells, and the loosely arranged cells are called as spongy cell, which is present below the palisade cells, and these 2 cells are collectively called as mesophyll cells. In these mesophyll cells, the chloroplast organelle are arranged along their wall. These are the chloroplasts which is arranged along their walls, so that they can absorb maximum amount of solar energy. So if we see the one organelle of the chloroplast, we can see this type of structure. This chloroplast is a double layered membranous structure. This is the outer membrane and this is the inner membrane. And inside the membrane there is a piling of sac, the sac-like structure which is piled up. So this piling of sac is called as granum. Granum means one piling of sac. If there are more than one piling of sacs, it is called as the granum. Grana are plural and granum is singular. So these granum are connected together by stromal lamellae. There is it is also called as fret. stromal lamellae connect the different granum together, and then the space which is filled with the fluid is called as the stroma, where the enzymatic action takes place. If we see the one sac, suppose this is the one sac, it is called as thylakoid. So granum is a collection of thylakoid. If it is piled up, the sacs are piled up, it is called as granum. And if we take the one sac is called as thylakoid. So the structure of thylakoid is also a double layered structure. This is the outer membrane and this is the inner membrane. And inside this, the lumen is present. So the thylakoid and the stroma, the whole photosynthesis is completed in two different reactions, the light reaction and the dark reaction. So the light reaction that takes place in this thylakoid and the dark reaction that takes place in stroma. Thylakoid, which is a membranous sac-like structure where the light reaction takes place, means thylakoid is responsible for the trapping of the solar energy, and by the light reaction, means the product of light reactions is a ATP. By light reaction, ATP is synthesized and NADPH is also formed. So in light reaction, there is the synthesis of ATP takes place and NADPH is formed. And in dark reaction that takes place in stroma, means in the matrix of the chloroplast, that by the dark reaction the glucose is synthesized. Glucose synthesis takes place in dark reaction, and this glucose is stored in the form of starch. So there is a complete division of labor in photosynthesis. Light reaction that takes place in membranous structure called thylakoid, and dark reaction it takes place in stroma, the fluid inside the chloroplast, and the products are by the light reaction forms the ATP and NADPH. They are also called as the reducing power. And the glucose is synthesized by the dark reaction. Light reaction is totally dependent on light, sunlight. So it is also called as the photochemical reaction. Photochemical reaction. And this dark reaction, it depends on the products of light reaction. Means this dark reaction takes place if the ATP and NADPH is available. So it is not correct to say that dark reaction takes place during dark or it is independent of light. After this site of photosynthesis, moving to the next topic, that is the pigments involved in photosynthesis. Various pigments are involved in photosynthesis. We can see this in paper chromatography. During paper chromatography, we see various types of pigments are there. Basically, these pigments are divided into two main groups. One is called the principal pigment and another one is the accessory pigments. Chlorophyll A is considered as a principal pigment because it is present in the reaction center, that we will see later on. And carotene, xanthophyll and chlorophyll B, C, D, these are considered as the accessory pigments. Accessory, as the name indicates, they help the principal pigments. They help the principal pigments in transferring the energy. They absorb different wavelength of energy, sunlight, and transfer the energy to the principal pigment, which is present in reaction center. Another function of accessory pigment is they protect the principal pigments also from photooxidation. Carotenoid. So in paper chromatogram, we can see the color of carotene is yellow or yellowish orange, and xanthophyll, so the yellow color, and chlorophyll B, it looks like a yellowish green in paper chromatogram, and this chlorophyll A is blue-green in paper chromatogram. So these are the pigments that helps in photosynthesis. In this video we had covered the importance of photosynthesis, the scientist, early experiments of photosynthesis, that you people have to develop some trick to remember, because the experiments are very important from NEET point of view. And next is the site of photosynthesis and the pigments involved in photosynthesis. In the next video we will cover the light reaction and the electron transport chain. Thank you.